welcome everyone. There has been a change in the schedule. So now it will be Rafael Sebastian from Universitat de Valencia presenting. It's really a great pleasure to introduce him. He's a very, very good friend that we've been working together for years and years in the community of modeling of cardiac electrophysiology. Everyone knows Rafa. I mean, he's one of the top players in this field, and everyone knows about his really fantastic work on modeling with great detail some of the superstructures in the heart. In particular, he's been working a lot in atrial physiology, modeling atrial fibrillation, but also ventricular tachycardia. But on top of this, he's a very good computer scientist. And so he knows how to code these solvers, how to play with these solvers. And lately, he's been also playing a bit like everyone with machine learning and how to combine the power of machine learning together with simulations. And to some extent, it's quite related to what we've heard in the previous talk on how to use machine learning to interpret whatever the amount of in silico results you can get from simulations. So, Rafa, thank you very much.
Thank you for the introduction, and thank you to the organizer for inviting me. Today, in this presentation, which is more or less in line with other talks that you've seen from Natalia Troyanova, Orazio Quarteroni, more related to simulation, and in particular today I will speak about electrophysiology and cardiac modeling. But over the last years we are starting to introduce or to combine the patient-specific models and the biophysical models with machine learning, as you've seen in other presentations. And the idea of this talk was basically to show some of the things that machine learning can provide to what we were already doing, and with the goal of translating some of these tools that we are developing to clinical environments. So just to start, a very brief introduction about heart arrhythmia. Well, you know that the main responsibility of the heart is basically to pump blood to the arteries, to the circulatory system. And heart arrhythmia, basically, it's a change in the heartbeat, okay, which is irregular. It can be too fast, too slow, or it can produce heart contractions, which are not very efficient. So here, for the ones who don't know, there is this movie, it's very famous, about a guy explaining what is sinus rhythm doing everything, so most of you have seen it. This guy moves the arms when the atria contracts and the legs when the ventricle contracts, so it's pretty easy to understand that everything is working fine now. When we have a problem in the activation of the heart, you see the guy starts to not moving the arms because there is some sickness, so it doesn't contract pretty well, the ventricles, okay? Now, there are other diseases like atrial flutter, where you see that the activation of the atria is very fast, but coordinated, and every now and then it passes the signal to the ventricles. So, atrial fibrillation. Okay, the atria is not working at all, and the signal can go to the ventricles from time to time, okay? This was an extra bonus, okay? And then there are other diseases not related to the atria, Wolff-Parkinson-White, where the contraction is not very efficient. Others in which, for instance, the atria is not generating the impulse of the activation, and then a pacemaker is needed. This is tachycardia, one of that we worked. This is very dangerous. As you know, when the ventricles are not contracting, the blood cannot be ejected, and you need to defibrillate the person. You don't have a long time for that. If you don't defibrillate the man, that is probably going to die of sudden cardiac death. And just to finish, you know, this is pacemaker. Some techniques like cardiac resynchronization therapy or pacemaking give a stimulus to the heart when the heart is not beating properly or it's not activating properly, okay? Well, I will pass this disc fibrillation. Guys, you can see it in YouTube. I'm sure you can find it. So, coming back again to the more serious stuff, this is very funny, but it's a very important disease. You've seen the presentation from Natalia. She was talking about sudden cardiac death. There are other diseases like atrial fibrillation that it's expected to be a pandemic in a few years because the population that suffers atrial fibrillation is increasing over years. And of course, there are other diseases like sudden cardiac death, are derived in 80% of times from ventricular arrhythmias that cause 50% of population death, of all death, included all diseases. So the thing is, what can we do to treat these arrhythmias? So there has been a very increased amount of technology that can be used to deal with this kind of diseases. You see here some integrated rooms that now hospitals use in order to treat this kind of arrhythmia diseases. In particular, cath labs, but these cath labs alone are combined with other tools and other rooms, like for instance, MR, CT, where you can get the geometry of the patient-specific geometry before the intervention. And as I was saying also, this is an EP lab where they can treat using radiofrequency ablation, or they can implant a pacemaker in the patient to restore the electrical activation. And in these rooms, they have pretty amazing technology. So they basically can navigate to the ventricles with a catheter. They can locate the catheter in real time, the location inside the heart. They can map the inner cavities using techniques like CARTO or NAVEX and know exactly what is the electrical activation in a point of the heart during the intervention. And they can perform, for instance, radiofrequency ablation. Okay? That basically what does is go into a particular location in the heart, and after finding where is the location of an anomalous activation like an ectopic focus, burn this area, okay? If there is an activation which is not following the normal propagation, it can create lesions inside the heart, like fibrotic lesions, that stop this propagation in such a way that electrical circuits is broken and the re-entry, the fibrillation, or the flutter stops, okay? I'm not going to get into detail, but of course there are other kinds of treatments that are the first treatment that you get when you have this kind of arrhythmias, which is based on drugs, not directly on ablation. With all that in hand, you've seen that still there is a high number of persons that developed and have problems with arrhythmias. Because in many cases, for instance, radiofrequency ablation does not work, and patient has to come back to a second surgery, what they call a redo. So you need, again, to have a second ablation or an optimization of the previous one. And this is basically summarized here, okay? This is a study from a few years ago from IBM that studied the correlation between the amount of money invested for different countries and the satisfaction of the population. And in principle, one can think that, okay, having all these tools and these techniques and these rooms in the hospitals, which are very expensive but probably provide better treatment to patients, people will be much happier. But the result was not very, very nice. You see in the United States, for instance, the investment, probably private, but the investment was huge, and the population was not very happy with the healthcare system, whereas in other countries it was much more optimized. And they tried to explain that with this plot, in which you see not only the tools that a person can have to diagnose, okay, but also the information that we know about the disease. So here, for instance, we don't know anything about a disease, and we don't have any tool to get information about this patient. So we do trial and error, because if we don't know very well the disease and we cannot obtain patient-specific data, we cannot treat it properly. And in the other extreme, we know everything about one particular disease, like we know everything about atrial fibrillation, and we have the best tools to guide the therapy in that particular person, and we can do personalized treatment. So I can get the geometry of the patient, I can have his electrical activation, I have the tools to reach the heart and do the ablation, and combining, I can provide a treatment to this patient that would be different to another patient. So it's more or less personalized. But the problem is that these tools that we are developing and these patient-specific tools have not reached the clinic yet. So we have several challenges to face. One of them is to develop predictive computational models of the heart or other organs that can be translated into clinical environments. So what I'm going to do is showing a few challenges, and then how can we tackle or how can we deal with this challenge. So the first is to develop these predictive computational models. Okay, what we want to do is the following. Well, this is an example in which that everyone can recognize. Everyone wants to know the weather in his area in particular. So you have here a map. This is Spain, and we have the weather forecast. So in order to know if tomorrow is going to rain in my particular area, I need to combine different types of information. If I don't have this model, what I can do is go into last year and previous year and see if it was raining in my country or if it was raining in my area, okay? And I can try to predict as a function of the past. But if we want to have a, let's say, country-specific, region-specific prediction for today, we need to acquire different types of information, which could be the first step. And we can acquire data from humans, from the heart, images, signals, etc. Then we have to combine all these different data into a model and then use this model with some biophysical tools, predictive model, in order to do a prediction if it's going to rain or it's not going to rain in a particular area. We want to do something similar with the heart and in particular for cardiac electrophysiology. So we developed this pipeline here in which we first want to build the anatomical model of the heart using either ex vivo experimental data or in vivo clinical data if we want to have a patient-specific model. We want also to acquire information about the electrophysiology. So this is this can be information acquired from the patient, or it can be also what is called population-based data from animals or for another population. Then we want to combine that in a finite element model, model some pathology that we are interested in, perform simulations to predict the outcome of some treatment, to plan the treatment, or to learn something about the pathology, okay, and take decisions. So we are going to start here with this part of how to build the anatomical model. For the ones who has worked already in this area, you know that it's pretty challenging to do the segmentation of the heart as a function of the modality in which you are interested. Here you have a 2D slice of a 3D stack of the heart, where you have highlighted different regions. And we can do the segmentation of all the slices of this stack to have the 3D reconstruction. And here you have the coronary artery, aorta, the atria, ventricles. And if we do segmentation over time, we end up with the geometry fitting. Okay, this is from a scan. The problem is that we are not only interested in the geometry. We also want to add information from different modalities that provide functional information. So at the end, this model has to be enriched with different data. Of course, we need scans to recover the geometry. That can be CT or MR. But we want, for instance, to add the substrate. We are very much interested in knowing where is scar tissue, where is fibrosis in the atria, if there are conduction channels that can produce reentries. And for that, we need to map all this data, again, into the model. If we have electrical data from the patient, let's say carto data, or other types of tools that can provide electrical information, we want to also add it. If we have information at the surface of the torso because we have ECG, we also want to add all to this model that later is going to be used for simulation of different things, like it can be mechanics, electrophysiology, electromechanics, fluid dynamics, etc. There has been a big interest in building such a model or these types of models. And if you look at the literature over the last years, this is a summary of numbers of papers published in the literature over the 50 or 60 last years. And you see that there is an increase on the number of papers published, this is PubMed, related with cardiac modeling and segmentation and construction of these models. From them, well, there are many that, let's say, are key models because they represented a change in the way that the model was built. Okay, so first, next, what we are going to try to tackle is this second challenge, which is develop automatic tools to be able to segment and construct anatomical models of the heart, okay, using machine learning techniques, as we see. And to finish this, we need to build this part, the electrophysiological models. So later we will see how we build these anatomical models. And we have to face three different questions that I think they have arisen several times over these days in this community. First, which type of model should I use? Okay. Second, what are the benefits and drawbacks of each type? Because we have many models that we can use for simulations, and we have to choose the best one, and what information is required for each of the models. So first of all, we are going to see the different types of models. some of the ones that they've been discussed over these days are biophysical models of the heart, okay? And in this particular case, cardiac electrophysiology. That models are multi-scale models that take into account the information at different scales. So we have the model of the cell, but not only the cell, but what are the molecular descriptions of this cell at the plasma membrane that allows the transmission of different ions from the inner to the outer regions of the cell, from the compartments. So we have cells with descriptions of all the ionic channels that are modeled by differential equation. When we put all this together, the difference in voltage between intracellular and extracellular potential is modeled by the typical action potential of the cell, okay? This action potential is integrated at tissue level using equations like monodomain or bidomain equations that model how this activity at cellular level is propagated through tissue, okay, generating a propagation wavefront. And then all this can be also integrated in a body model that can simulate the electrical activity that goes from the heart to the torso and produces the electrocardiogram, taking into account all the geometry, the patient-specific geometry of the different organs, the conductivities, etc. So if we want to solve such a model, we can go to different levels of detail. The first level of detail, which is the most detailed, is the one in which we model each single cell of the heart. We model the tissue as it is, but the problem is huge because, as you can see, if we model the heart using cells of 100 micrometers, which is typically the size of a myocyte, we end up with 423 million cells. For each one, we have different channels for the ionic models, and we end up with 1.27 billion equations that we have to solve every time that we solve this. Assuming a normal computer with one core, okay, to simulate 1 minute real time, we need 500 days, which this is something that we cannot wait, okay, to do a simulation of 1 minute for one patient. Even if we rely in high performance computing, this level of detail is far from what we can tackle here. So what usually we do is we have the hearts and we do a discretization which is much coarse, okay? So we end up with 4 to 12 million elements per models, which is 3 to 6 million cells probably. Solve every 10 microseconds. We have this model of 30 more or less ordinary differential equations, and to solve one of these models we need around 4 hours using 64 cores, which is something much closer to clinics but still complex to translate. Then there are these other models which are usually called simplified models of the heart, in which we have really coarse meshes between 10 to 20,000 elements, also 10 to 20,000 cells. We solve very simplified models like the Michel-Shepard that has only 4 variables. And with that models, we can simulate in a matter of seconds a heartbeat. So we have to decide between, as a function of the application, which of these models we need to use, and basically depends a lot on what is the question we are trying to answer, okay? So detailed versus simplified, which type of model should I use? As I say, depends a lot on the question and the application of the model. What are the benefits and drawbacks of each type of model? Well, with the detail, we can complex and test hypotheses that cannot be answered with the simple one. For instance, if I want to know how a particular drug is interacting with a channel in the cell and how this affects globally to the heart, I need a detailed model because the other ones do not consider the particular ion channels. But if I want to do some kind of simulation to guide an intervention for the doctor that just needs to see how the heart activates, but I don't need to go into too much detail, a simplified model can be easier to implement and has another advantage, which is that with a few parameters, I can personalize a model, whereas in the other one that has so many parameters, it's pretty difficult to personalize. And what information do I need? Well, for both of them, I need data from the patient, but for the detailed one, probably I need ex vivo data. For instance, I need data from patch clamp to model all the channels in the cell. I need histology data to model all the details of the heart, and this is not always available, and it's not patient-specific for sure, okay? So we have with this a third channel, which is to develop tools based on computer simulation that helps in diagnosis and therapy planning. Okay, so these are the 3 different challenges that I think I want to present today. We are going to start for the first one. Challenge 1 was to develop a predictive computational models that can be translated into clinical environment. For that, because I want to focus a little bit on machine learning, I'm going to show examples in which machine learning techniques can help to perform these kind of things. Okay? I know that probably most of you know about machine learning. This is a very short background. Okay? For the ones who has read less about it, the idea of machine learning is to train a computer to do something that humans do very naturally. So to use data to train how to do some specific task, but not specifying the task by a model, okay? So combined with other tools like big data, it opens new ways for research. Basically, it has a lot of impact in natural language processing, but also in medicine can be used to take decisions based on previous information that was used for training the model, okay? The broad division of these machine learning algorithms, it's in 2 groups: unsupervised learning, supervised learning, and reinforced learning. Okay? Each of them, here you can see a few applications for them, but this is much more broader. This is just a very, very short summary. I circle one of the ones that can be interested for us. So for instance, for image classification, that I'll show you later an example, supervised learning is working pretty well. For diagnosis, it's also used in order, for instance, to differentiate different types of rhythms in the heart. Here I can see very well, but it's estimating life expectancy, okay? And here for learning different kinds of tasks. So let's see a little bit more supervised learning, okay? In supervised learning, what we want to do is the following. We have here an algorithm, and we have our database of data here. The data is labeled. It means that each of these points represents some data, which can be a heart, or can be an ECG, or can be something which is labeled. I know already to which class it belongs. For instance, if these are ECGs, I know exactly if one it's an ECG of a patient with ventricular fibrillation, another is in sinus rhythm, another is with atrial flutter, and I have these labels. All this information is passed to an algorithm that has to learn to differentiate, just looking at the data, about these labels, okay? So we end up with something like you have here. We have the different samples, each of them with a specific label, and we need to find this function that can be nonlinear to differentiate between both of them. We train the model, and then when we show new information to the algorithm that has not already used for training, the algorithm has to classify as one type or the other and give me the answer, okay? So this would be supervised learning. In unsupervised learning, we start with a database here in which the data is not labeled yet. So I know these are many different types of ECGs, but I don't know which is the label that they have associated. So basically what I want to do is I give to the algorithm this information, and I want that the algorithm clusters in groups this information, like you have here. So I have all this data, they can be ECGs, and I want that the algorithm tells me how many, well, there are 2 questions. We can ask how many groups, or we can tell, we have 3 groups. I know that there are 3 types of ECGs. I want that you split them, okay? So in that particular case, it splits the groups in these 3 different ones that later can be used for training or for finding also to which group belongs a new signal that I obtain later, okay? And then the 3rd type, which is in fashion now, it's starting to be used more and more, is what it's called reinforced learning. In reinforced learning, we let the algorithm to take decisions about results or internal decisions that the algorithm is taking. For instance, we have all this data, and this is a system that basically takes a decision, and then there is a function that takes if this decision was good or bad. If it's good, we reward the internal classification. If not, we put like bad marks. Like a friend of mine working on Siemens used this algorithm in order to segment the heart. So basically, they let the system try to segment the heart, and then if the heart is well segmented, they say good job to the agent. So he learns that this is the way to segment. If it's bad segmented, they say, bad job, don't go this way again. So the algorithm by itself learns how to perform the segmentation without an explicit modeling of the contours or the definition of the regions of the heart. And I want to show here an example in which supervised learning was used for classifying different types of signals. This is the challenge of computing in cardiology of last year, 2017, in which a database with 5,000 ECGs was provided. They were labeled by a doctor, so we knew exactly if there were normal ECGs, atrial fibrillation, other rhythm that can be anything, and noisy samples. The thing is that we have to take these 5,000 signals, we have to train something, and this algorithm has to be able to recognize signals that hasn't been observed. Okay? These were the results. The winner was Tejero et al. With 83% of accuracy in the classification. Okay? He used machine learning. Basically, he used a deep learning technique. Okay? That was the following. Basically, one of the good points that they included to get this extraordinary result of 83% is that they did not believe on the data that was labeled from the beginning. So instead of taking the data and training the algorithm, they took all the data and they relabeled it. Why? Because doctors are persons and they make mistakes, and there were some data that was mislabeled. And that was introducing a lot of bias in the algorithm that was not learning properly. So they take the database, they put some rules, they checked everything, and they discarded all the ones that were incorrectly labeled. After this, and this is what they call adaptive interpretation. So a doctor tells them, in order to recognize this, I look at this, and this. Like P wave has to come first, then QRS, then the signal has to be this, and this way. So they analyze all the data, and as a function of that, they relabel and get what they call global parameters from the data. So parameters that appear all over the sample. Then they have a second part, which is this per bit feature. So instead of looking at the whole sequence, they look bit per bit, and they develop this pipeline in which they use multi-layer perceptrons, and this LSTM, which is in fashion now, is long short term memory, and basically it's a deep learning, let's say, building block in which you can learn things that you pass and has temporal information, let's say, okay? When you want to learn something that includes temporal information, LSTM work pretty well because there are neural networks that has memory, and then they can recognize patterns, okay? So combining all these tools here, at the end, they can predict or pool between two different information, that one that comes from global classification and the one that comes from per bit classification, and they decide which of the 4 classes it belongs. You have to take into account that this 83% also suffers from mislabeled test set. It means that if the doctor has labeled a test set that it's incorrect, some of the samples that the algorithm probably has classified properly will be considered incorrect because the test set is not properly corrected. Okay? So when you train a model, it's very important that the data is curated and it's well labeled. Okay? If not, the algorithm, instead of working well, probably you introduce more bias to the result. The second challenge was to develop automatic tools to be able to segment anatomical models of the heart. Well, there has been a lot of research in this sense, okay? The idea is that we need to segment different modalities to identify structures, right, left ventricle, endocardium, epicardium, everything, okay? Not only from CT, which would be the most obvious, but from MR, ultrasound. And we want to do it as automatic as possible because this is very, very time consuming. The ones who have suffered the manual segmentation of the heart, if there is any here, will know that this is very time consuming and a bit frustrating sometimes. So the initial or the classical techniques used image-based or intensity-based algorithms. For instance, the ones that are based on the histogram for segmentation, region growing, active contours, level sets, which are one of the most sophisticated, can achieve good results, but depend a lot on the quality of the image. Okay? So if we have an image that is pretty well well, the signal to noise level is very high, you can see the contours, okay, they work more or less well. But when we have problems because the contours, for instance, are lost in a specific region of the heart, or for instance, because some of the samples at different heights are not very nice, it's very difficult to do the reconstruction because we don't have any information to be based. So the idea is, why don't we use something different than only the intensity level? So let's use model-based segmentation. And the model, it's something related with machine learning as well. The idea is, let's learn what is the shape of a heart, and then, knowing what is the shape of a heart, let's try to take a heart that can be deformed to adapt to an image, instead of segmenting the image completely without any a priori information. So what I'm going to show now, it's this model-based segmentation. Basically, it's called active appearance models. They have been in use for several years already. And the idea is that we are going to build an atlas of the heart that basically it's a point distribution model of its contours that can be deformed to be adapted to any image that we want to segment. In order to build this active appearance model, we need three different things. The first one is a shape model of the heart, what we call the atlas. Then we need an intensity model. You will see later, because it's not the same to segment an SPECT image than MR than CT. Okay, grade level intensities are different and we have to adapt to them. And then we need a matching algorithm that can allow us to fit this mesh into this particular image. Well, the first step in order to build such a model is to get, as usual, a database with many hearts, as many as we can. Okay, so we have many hearts, and we have to register all of them. Probably all of you know what is registration, but for the ones that doesn't know what is registration, basically we have two images, and we want to put them in the same reference system, and we want to deform one of the images so that it matches the second one. Okay? So we need some similarity measures between the image and an objective function that is the one that we are going to minimize to make both images to overlap or to match. Okay? And then we have to do a transformation on one of the images. The transformation can be linear, in which we just rotate, move, and change the scale of the image. Or it can be elastic or non-rigid. In that one, we deform actually the original image to match exactly the second image. Like, for instance, you have here an example. This is the target image. This is the moving image. Is an image of the same area, but taken from different angles. So what we are going to do is to take this image, rotate it, and scale it so that both of them fit exactly. Okay? We are going to do the same with the heart. So now, well, before reaching the heart, we are going to use a second tool, okay, which is the point distribution model. Okay, and for that example, I'm going to show you with bones what does this mean. If we want to build the atlas, imagine that you have hands and we're interested in doing a model of one of these bones from the finger, okay? So what we need to do is first segment all the bones and add all the landmarks in different specific places. Then we put all the bones together, okay? Then we are going to do an averaging and a matching of all the landmarks. And from this, we can learn all the different shapes that a bone can have, using, for instance, principal component analysis to keep only the most important modes of variation and then building a model of the bone. With the heart, we do something similar. So we take all these hearts, let's say this is a database with 100 hearts. We take one of them as a reference, which is going to be this heart here. Then we register all these hearts so that all of them are aligned, okay, because each heart can be rotated or it can be displaced, it can have different sizes. So we actually do all the transformations. Then after all of them are aligned, we do an average of the heart. So we have an average here of all the transformation, and we have the average with non-rigid transformation. And this average heart with non-rigid transformation is what we are going to call the atlas of the heart, okay? So all these images has been registered to this reference image, okay? And in here, we have encoded all the transformation that lead from the original image to the atlas of the heart. Now, we take the atlas and we perform a very detailed manual segmentation of the heart. So the atlas is very well segmented. And then from this one, we undo all the transformations that we have carried over these lines. So we have, again, all these original models, but segmented, okay? Because first here we do the transformations, we do the segmentation, and then inverting each of the individual transformations from the atlas, we obtain every particular mesh segmented. With this every particular mesh segmented, we are going to train our model to do the principal component analysis. So summarizing, we have the hearts, each one is different. We align all of them. We perform the transformation to have all of them registered to the atlas. Then we do the segmentation of the atlas. We take all the transformation and we undo this transformation. So we have the original hearts landmarked, and then we do the principal component analysis. So in this part in which we have all this landmarked, we have something like this. Okay? We have many hearts. We end up with this. All of them are more or less aligned, everyone which is specific landmarks. And then we do principal component analysis, and from this we obtain which is the representation of each of these hearts in the principal component analysis space representation. Because we don't want to take all the degrees of freedom of the model, because it's huge, and there are some difference between hearts that we are not interested in. We're interested only in the main variations. What we are going to do is to restrict all the information that we use, using here the auto vectors and well, all the information that is important for us. So imagine that this is each of these points is the population of hearts that you have. Each heart is represented by one of these points. We are going to restrict with principal component analysis to this area that is yellow, meaning that for us, everything that is here, every of these points, it's a heart. Everything that is outside here is not a heart. It's very similar, but it's not a heart for us. So in order for us to be a heart, the covariance matrix has to end up in one of these, well, inside this ellipsoid. Okay? If we now take hearts along this line, which is the principal component, we have different hearts. This point is this heart, this other point, this second heart, this last point here. And you can see that the difference between one and the other is the size of the atria or the size of this ventricle in yellow. So every point here is a heart. Everything outside, it's not. Using this technique, we build this heart. Okay? This is built from 100 subjects in 15 cardiac phases of the cardiac cycle. This is a point distribution model of the heart that basically corresponds to the average heart. With this, we have something that it's interesting, which that can be very useful for simulation, and is that we have an average representation of the heart, and then providing weights to this vector, we can obtain new hearts. New hearts that all of them end up inside this yellow area, but we can have a large population of virtual hearts to do whatever we want, modeling, shape analysis, or whatever we're interested, because we have learned what is a heart from our database, and then we can model different variabilities that are realistic. Okay? If we want to use it for segmentation, we have to learn also the intensity levels at each point of this mesh. So if I want to fit this model into this 2D, let's say, plane at different heights, I need a matching algorithm. So in order to do that, for each of the points in the contour of the heart, I just, for instance, look here. This is the gradient. That is this representation here, how the intensity level changes over this line. And what I'm going to do is to move this shape, which is the average heart, towards the contours of the heart, because I want to deform it to that particular case. So let's imagine that we have these different 2D images, okay, from a 3D scan. I just put there my atlas. It doesn't match at all. You can see that, well, here it matches more or less, but it doesn't match very well because it's not following the contours. So basically, I look at the gradients and I deform in this particular area my algorithm, my shape. Okay. If I do it for all the cuts, I will end up with something like this. Okay. I've been dragged by it, but if you see this, you can recognize pretty easily that this is not a heart. And the model also recognizes that it's very far from what we consider a heart. So we basically project into our space this model, and the closest one is here, and is this heart, which is the closest to this one that it has the shape of a heart. And then I can do this iteratively until I converge to a model that doesn't change that much between one iteration to the other. If we put this to work, we start with, you see, this is an image of a heart in at different planes. This is the contour. It's pretty far from the borders, as you can see here. I press play. It starts converging until it fits pretty well the ventricles. So we have used here a priori information, which is the shape of the heart, in order to fit the particular data of a patient. And well, this is not magical. This has some problems. If the gray level intensity is not very good, of course, the system is going to have problems in order to recognize the contours. If the heart is very different to the ones that I use in the training data set, I'm not going to allow the deformation, and then I'm going to have a suboptimal segmentation and not the real one, okay? One of the good points of having this kind of atlas is that when I finish the segmentation, I can have already propagated the labels of the atlas. Here you can see the endocardium, the epicardium, atria, and ventricles are automatically propagated. So I can split these different regions. I can encode also information that is functional. For instance, if I build also in the atlas information about the movement of this wall, okay, I can try to see if the wall is moving properly. Okay, so here you have the movement of this. This is the septal wall. Okay, this is an ultrasound image where you see the ventricles. This is the septal wall, and here you have the region that represents the normal movement of the septal wall. If we now take the atlas of the heart, we segment all the cardiac cycle, and then we plot this blue line. You can see that it's pretty out from the average or what we have learned already. Okay. Here, in this 2D representation, this will be the normal movement of the wall, and this would be an outlier. And it can be very helpful in order to detect abnormalities in the motion of the heart, because this information is encoded in the atlas. Okay. So in summary, using based models that are built from data learned from a training set, for instance, can be very useful for segmentation and to include structures, functional information, and on top of that, it can work for different modalities. It's a compact representation that can be also used for cardiac segmentation and for generation of heart populations. Okay? And also, some people has used this kind of models for quantification or for phenotype analysis of different patients. But this is not in fashion. I mean, this is something that has been used for the last year. But what everyone is trying to work with now, it's, as you know, with deep learning. Okay. As a background, well, deep learning is based on neural networks like this one. Okay. This is a typical neural network with an input layer. Each of these circles is a neuron. This is a hidden layer, and this is the output. Basically, when you work with this type of networks, what you have to do is give weights to all these connections between different layers in order to obtain the result that you desire. As an example, if you input all these images with particular labels in which you have trucks, sheep, horse, frogs, dog, okay, to one of these networks, your goal is that every time that you give a new image, in the output, you have a high probability or the highest probability to the label that you have used as an input. So combining networks that has lots of neurons like this appear deep learning, and one of the networks that is also being used a lot for imaging, it's convolutional networks. Okay? The convolutional network reduces or simplifies a little bit all the cost of finding all the weights for all the connections between neurons by doing some pooling of information. So for instance, instead of connecting each of these pixels with all the pixels in the next layer of the representation, what we do is we take an area and we work with all this area, and all this area is going to activate only one neuron in the next layer, instead of activating a full mesh-connected layer of neurons, which makes things much easier to work. And on top of that, what we do here in this specific square of the image, we can replicate it everywhere in the image. So we can learn the weights for one particular area and reuse them a lot inside this. So putting all this together, some researchers over the last years have started to develop connections or pipelines interconnecting these kinds of networks. For instance, You have here a convolutional network that was used for segmenting the blood pools of right and left ventricle here, from here to here, in which you have all these layers. Other people uses the convolutional network not for segmenting directly the pixel, but to initialize a shape model like the one I showed you before. So they combined the shape model of the heart that they have learned with machine learning that basically uses a classification to say what type of pixel is seen in the image in order to perform the segmentations. So many papers are expected on that topics in the next years, and most of the challenges that are now, like the MICAI challenge and other challenges that are now being published, usually the winner you see that uses this kind of techniques to process the data. The third and the last of the challenge was to develop tools based on computational simulations to help in diagnosis and therapy planning, which was the last part of our big pipeline. So we have worked mainly now in this anatomical model, but I want to do simulations that can be used for decision, for the doctor to plan and take decisions. And the example I want to show you, it's the inverse problem in cardiology. Okay? In the inverse problem in cardiology, we are interested in knowing what's happening in the heart from information that we collect from the torso surface. So a patient comes to the clinic, we put all these electrodes, usually it's a vest like this one, in which I can measure the potential everywhere on the surface of the torso, not just in the precordial leads. That would be the standard ECG. And from that data, I want to solve what is called the inverse problem in electrocardiography and to see what is the potential at each time step in the heart. So this is a a non-invasive technique in which I estimate from the torso the potential in the heart. This is an ill-posed problem, so you need regularization to solve and some mathematical tricks to obtain something with meaning here in the heart, and it makes the problem a bit complex. In order to solve this problem, you need also the geometry of the torso and the geometry of the ventricle or atria, where you're going to inverse the problem, because the orientation is pretty important in order to do the reconstruction. So what we did in our lab was basically build a full heart model. So we reconstruct the surface of the heart. We included patient-specific information when available, like a scar for magnetic resonance, some other structures like the Purkinje network that was modeled mathematically because we cannot obtain it from in vivo measurements. So we built all this model. Then for the atria, where we could not include the fiber orientation, we study basically how was the fiber organized using histological data. So from this histology data provided by a friend of us, Damian Sanchez Quintana, we mapped all this into our atria, like this, like you can see here, where we have divided in 50 regions the atria, and we have incorporated the fiber orientation in each of them, okay, to account for the anisotropic propagation in the atria. So all this is incorporated in a torso model. So this is the torso model that includes different regions. We have here the lungs, bones, the liver, and also the heart and the blood pool here as well. Okay, so with all this together, we can simulate what's happening in the heart and how it propagates to the torso. So we here used detailed models of the heart, and we incorporated a model which is called Kortemann. So Kortemann is a model that can reproduce quite well the action potential in the atria. But because in the atria there are different regions, what we did was to differentiate different regions of the atria and have region-specific atrial potentials, giving this heterogeneity in the heart. We also here model the conduction velocity in each of the regions of the heart, and then we perform simulations using the monodomain approximation. So we could have activation of the atria that then we compare with the literature to see if the activation was properly done, okay? And then we propagate all the activation of the atria to the torso, and then we obtain the electrocardiogram. And in particular, the P wave, which was the only part we could obtain because we were only simulating atria, not ventricle. So with all this, we can simulate normal rhythm, but also flutter, tachycardia, and different types of fibrillation. This is very time consuming. We cannot build this for every patient that comes to the clinic, and we cannot simulate very fast different rhythms because each of the simulation takes ages and building the model takes weeks or months. So our idea was, can we use this model to train a system based on machine learning that can predict the outcome or can classify different types of rhythm automatically without building the whole thing? So we started with atrial tachycardia. What we did was we checked the literature where there is information about the electrical pattern obtained at torso level, stimulating at different points of the atria. So here you have different locations where a doctor is stimulating, and then this here, it's the pattern that is obtained at torso level. So we did exactly the same in our model. We stimulate in the same locations, and then we obtained the pattern here, which basically is, it was, the color represents the integral of the P wave in different locations of the torso. So we did it for many locations that were published in the literature, stimulating our atria at different places and watching what happened. And as you can see, this is our simulation, and this is what is obtained in the clinics. So you can see the green part match and the blue part more or less. This is stimulating in another location of the atria. So we observed that our pattern resembled pretty well what was obtained in the literature for the torso. So we developed a pipeline in which we put many ectopic focus all over the atria to simulate activation that were ectopic. And we obtained our torso representations, or the propagation to the torso. From there, we map a few locations, okay, to go closer to the clinic. We map basically 256 locations which correspond to these patches. And then we provide all this information to a system that was doing unsupervised classification. Basically, we did k-means, which is the easiest one and worked pretty well with our data. So our data was not classified, was basically all these patterns. Then after classification, it divided our samples in different groups. And we use this group as a label, okay? So we have, as a function of the number of classes that we are interested in analyzing, here we have them like a color, well, based on a color, okay? We have divided our samples at torso level in different groups, and then we use them for training. After all this, we also selected, and I'll show you the results later, which were the locations in the torso that provided the most information in order to identify to which class belongs each of these models. And finally, we train a support vector machine model to see if we can predict the result of the outcome, okay? The support machine, basically, what does is, once the population is labeled, okay? Here we have two classes. It tries to find a function that splits with the maximum distance to both groups, the two populations, okay? And here you have several examples in which a lot of samples are divided in 3 different groups using a support vector machine. We use a support vector machine with radial function, which will resemble something like this. So our contours are something like this, okay? So after this, we developed stratified cross-validation, in which we have all our samples, which are the torso samples. We divide them into groups, a training set and a test set. Because this is stratified, the test set has to include representatives of the different classes in a proportion which is similar. And then with different folds, we carry out all this cross-validation and we average the results. And these are the results that you can have here. So in the horizontal axis, you have the number of features, so the number of electrodes we use in order to classify. And in the vertical axis, you have the accuracy of the model. So when you use 2, 4, 8, 16, and 32 electrodes to classify the arrhythmia, we have a very poor accuracy here, but from 32 electrodes on, we have more than 90% accuracy. That means that if you show the system an activation, the map at the torso level, it can predict the location of the ectopic focus in the atria, okay, before the intervention, with 90% accuracy. That's very helpful because doctors, if they acquire this map, the system is going to tell them that you have to go probably to that area of the atria because in that region probably is the ectopic focus, okay? And there is a secondary zone that it's the one that can be predicted. So in conclusion, the development of accurate biophysical models of the heart that can simulate pathophysiological scenarios can be very useful for optimizing treatment planning and delivery. Combined with machine learning algorithms, they can aid in reduced subjectivity, and also using the models as we have used them here to train not as a tool to predict the outcome, but to train a model based on machine learning can be useful because, as you have seen, the pipeline that we've developed doesn't require to do new simulations. So it gives you an answer in a matter of seconds or less than seconds, compared with making the whole patient-specific model and trying to do the full simulation. And with this, finish.
Any questions from the audience? No, not really. Rafa, to some extent, you are using machine learning to run a lot of simulations and try to better understand whatever physical phenomena in the heart. But you haven't mentioned applications of this for improving the image processing side or the data processing side. Do you think it could help on this sense?
Yes, well, depending on what you mean by processing data. For instance, in the first example, the group that was developed the algorithm in order to classify the different types of rhythms, using the software or the approximation, they could define bad labeled data. So this is because probably different doctors have different criteria and they have labeled the data in a different way.
This is quite tricky, right? I mean, because you are kind of imposing other criteria for the labeling. No, I mean,
well, an homogeneous criteria, because there were different criteria for labeling the same type of data, and that's the problem. Because there were different people doing the same things, and they were not close together.
But you can do it in a limited data set, no? I mean, this relabeling...
Yes, probably yes. Well, this was a 5,000, but yes, it's time-consuming.
And so you see more and more, I mean, this combination of running a lot of simulations as a way to train these kind of models and then use them for processing real data.
For instance, yes. Well, this is the approximation we want to follow because we know that it's very time-consuming, complex, and it requires some specific skills to build the model and to do the simulations, and this is something that is not usually inside the clinical environment. So in order to translate something right now, you need to give them tools that are easy to use, fast, and can give an answer in, let's say, their times. So simulation now, it's a bit far from that. And, but it can be used for performing things that we cannot get the data of different ectopic focus everywhere in the atria because we cannot get a person into the EP lab and stimulate there. It's very invasive. And the only way to obtain this data is using a simulator. Mm-hmm.
Yeah.
Thank you. I have a question. Why did you choose a support vector machine and not a neural network to do your classification in the...
Yeah, the problem was not very tough to differentiate different. As you can see for the results, it was not a very difficult problem. And SVM worked fine. If the problem would have been more complex, maybe we will use something more specific, more advanced, let's say. But as you see, with SVM, it worked pretty well. Now we are including a lot of noise to the samples and fibrosis in the atria, so it's much trickier to do the classification, and probably we will explore different ways to do the classification.
And do you expect a similar pipeline to work for ventricular arrhythmias? Because there can be...
That's something we are trying to do right now, so we don't know. But it depends on the repeatability of the data. I mean, if unless you have a huge database, if the cases are very different and are complex to classify, I don't know if it will work. But I've seen already papers using similar techniques in order to differentiate where an ectopic focus comes, but only dividing the heart in 17 regions instead of everywhere in the heart.
Okay. And just one last thing. It's because of course the morphology of the P wave and well later in the QRS, it can change if you have some underlying pathology, like if you have ischemia, HCM or whatever. Do you take that into account when you model and train, or you just assume normal models?
Well, the thing is, for some pathologies, you cannot obtain the same index we are using. For atrial fibrillation, for instance, you can obtain the integral of the P wave because there is no P wave. But if there are some, let's say, pathology that alters the geometry, we use exactly the same pipeline because we consider that the model has to be able to differentiate these kind of things.
But you think that the prediction will be the same even if you
no,
it will.
The result will be much poor. Yes, for sure.
Okay, thank you very much.
There is life in machine learning beyond deep learning. No?
Of course.
I mean, there was, I think, a recent paper where talking about deep learning, but in the supplementary material, they were showing that regularized logistic regression were having the same results as accurate as deep learning. Obviously, it was in the supplementary material, but I mean, we need to take into account this. Okay, so thank you very much, Rafa, again.